entering into the new chapter called lac operon. The lac operon denotes lactose operon. We know very well lactose, a milk sugar, which is highly prone for getting breakage. When there is a utility, when there is a utility, since the bacteria requires a specific lactose, okay, when there is no glucose at all. For example, I'm a bacteria, I'm not having glucose, I'm not having fructose, I'm having only lactose integration medium. So I will use lactose. So if I am having glucose also, lactose also, then I won't touch lactose. I'll touch only glucose. So the bacteria behavior sense changes depending upon the nutrients availability. I'm not having glucose at all. In that case, I use lactose. So that lactose utilization potential of the bacteria is controlled by a set of genes. Those set of genes are controlled by other set of genes. Those type of genes, how it is governing the lactose metabolism, lactose breakdown, that we are studying under the lac of rod concept. One more time, I'll repeat. Keep the concept in your mind in a very simplified way. Lactose breakage is mandatory for the bacteria to survive. If the bacteria is not having glucose, this is taken care by the genes, which is going to code for the metabolic enzymes. That metabolic enzymes are coded by the lactose controlling genes of the bacteria, and those genes controlled by other group of genes. So this how one gene is controlling other gene, and how the controlled gene is governing the lactose metabolism. Everything is studied under the lactose metabolism, under the lactose operon concept, right? So we'll be entering the new important subtopic on lac operon, right? So there is a beautiful example for the regulation of the gene expression. For example, uh, I'm going for one very good Western song, right? So, or else I'm going for some Carnatic song. So, when I'm going for rehearsal, okay, my master is saying something. This is the way you need to increase your tone. Okay, and the other person is saying, okay, here you need to reduce your speed in this junction. Other person is saying, in this area you need to increase your speed. Okay, here you okay like this. What everybody are adjusting their vocal chord vibration. So, like this, the gene expression. What do you mean about gene expression? Okay, for example, the gene is making the DNA, gene is making the RNA, gene is making the protein, and it's called gene expression. For example, if a person is coming to our house, okay, he is one of our close friends. Okay, when the concerned person is coming to our house, we are very happy to receive. For some person, why he came, we don't know. So, like this is what we don't want to see his face. So, some we are agreeing, some we are not agreeing. Right? So, accordingly, our face changes happens like this in the same way. The gene expression, sometimes it happens very fast, sometimes it happens very slow, which is controlled by other group of genes that is influenced by so many parameters, not only environmental parameters and even the biotic parameters itself, right? So now we are entering into Lacaron in detail. The regulation of the gene expression. If we're talking about the regulation of gene expression at the transcriptional level, the primary transcript where the genetic code is transcribed from the DNA. See, listen very carefully. Primary transcript the word. I'm saying for immature RNA. See, here the sentence is there. What is the other name of primary transcript? Immature RNA. Immature RNA is also called as what? Heterogeneous RNA. So those words are not there in the slide. Okay, if I'm coming to the class being a teacher, I should have the multiple vocabularies so that what I will guide you properly. The same thing for you also. What we taught in the last class, with the preparation coming to this chapter, then you can tap the message much more in the advanced version than the existing one, right? So last class division is very important. And the second thing is, whenever you're saying multiple terms for the same term, which is not there in the slide, you need to write it down while listening to this lecture, right? Primary trans or immature RNA or heterogeneous RNA is impotent or not capable enough to perform the protein synthesis from that structure. You need to get modified furthermore to become a mature RNA. For that to happen, so many number of enzymes are there, which is taking the entire event in a more appropriate way, right? So now we're talking about introns and exons are part of the immature RNA. If I'm an immature RNA, if I need to become mature RNA, then what are the things I should take it away from my body? Okay, introns should be removed away from the body. If the introns are removed away from the body, then only what? The mRNA is in a position to carry the protein synthesis process. Right. Now we are coming for the transport of the mRNA. We know very well the mRNA, once it is getting synthesized in the nucleus, it is going to the cytoplasm to take the responsibility of protein synthesis in the ribosome. Okay, for example, here I'm the mRNA. I take the message from the DNA, I'll go to the ribosome, and then I'll govern the ribosome with the way to do the protein manufacturing. This is the way to do the protein synthesis. Like this, who is governing? mRNA is governing. So mRNA is found in the nucleus, right? This is a message we can understand in lack of doubt. Right? Any one of you have any doubts? Just I'll see the Q&A section. Please let me know. I'm going to help you. Okay? Any one of you have any doubt? Okay, Vaishali, no? Any doubt? Okay, any one of you have any doubt? Please let me know. Right? Now, we are entering into the next important concept in the translation is nothing but transport of the mRNA molecule from the nucleus to the cytoplasm. So the transport of the mRNA molecule from the nucleus to the cytoplasm that we already told to you, okay, it is not going just like this. mRNA wears cap and tail and then it goes. Why? Because when it is getting migrated from the nucleus to cytoplasm, so many negative elements like enemies are there to break the RNA. That is why the protective structure, the cap and tail is added to the mRNA, okay, while migrating from nucleus to the cytoplasm, right? So now we are entering the next concept called in the lack of power. The beta galactosidase enzyme is synthesized by the E. coli, right? How this enzyme works? Beta galactosidase enzyme works only when the lactose alone is there in the absence of glucose. If the glucose availability is there, then this beta galactosidase will not work. Beta galactosidase will not work. For example, I will say to you here, when you are angry or when you are having the full temper, we can't control our body activity, right? So, okay, we lose our behavior sense. Okay, like this, the beta galactosidase will not work more appropriately if the glucose is present. Okay, if the glucose is zero, only lactose is there, then the beta galactosidase breaks the lactose into glucose and galactose, right? So, lactose is not present in the environment. Then the beta galactosidase enzyme breaks the lactose into glucose and galactose, right? So the lactose ingredients are made up of glucose and galactose is not there in this slide. So whenever we are saying what are all the extra points we are saying, we need to write simultaneously in the note. Okay. Now, if talking about in the gene expression, we already told you what is about gene expression. The DNA is making the RNA, RNA is making the protein. That is called gene expression, right? The gene expression is controlled by many parameters, as I already told you. One is activator, another is repressor. So activator, repressor are formed by the respective genes. Activator gene makes activator. Repressor is formed by the respective genes. For example, if the repressor protein is present, then this entire lack of protein will not work. So lack of protein will not work means in the sense what? Beta galactosidase will not work. If the beta galactosidase is not working, what will happen? Lactose is not broken. Why the lactose is not broken? Why? Because beta galactosidase is not working. Why beta galactosidase is not working? Why? Because the concerned enzyme is becoming inactive in the presence of glucose. That is why this enzyme is not working. Okay. If the glucose is removed, this enzyme will work. So instead of saying this in a sentence, what they will do, they will ask the question this way: lack of prawn switch on and switch off mechanism. Lack of prawn switch on and switch off mechanism is because of what are the parameters? Like this, they will say lack of prawn is switched on because there is no glucose. Only lactose is available for the bacteria to survive. Lack of prawn is switched off. Why? Because glucose is available along with the lactose. So beta galactosidase will not work. Lactose metabolism will not happen. In this particular instance, bacteria will take only glucose. Bacteria will take only glucose, right? Now we are coming for the next phase of explanation. Before that, you understand one concept. This beta galactosidase enzyme breaks the lactose into glucose and galactose. This is one information. If they're asking the same question in the case of 2 mark or 3 mark, we need to narrate a little bit extra. What extra? Lactose is broken into allolactose, and then we got the glucose and galactose. Right? Now we are coming to lack of prod. In the lack of prod concept, what we need to understand is the Francis Foucault, okay, and then the Morard, the two great scientists, they explained the lack of prod and they got their own prize. So if you're talking about the polycystic mRNA or structural gene, when the moment whenever they're saying the hidden meaning behind that is this polycystic structural gene makes a multiple protein. If I'm saying monocystic mRNA, it makes single protein. If I'm saying monocystic mRNA, single protein. If it is polycystic mRNA or structural gene, multiple proteins are formed. See, tryptophan operon, arabinose operon, histidine operon, valine operon, all these are all the different type of operons which are working simultaneously. So operons are made up of first of genes, which controls other set of genes. The other set of genes controls the enzyme formation. The enzyme formation controls the lactose metabolism, right? Lactose metabolism is controlled by enzyme. Enzyme is controlled by one set of group of genes. That group of genes are controlled by other set of genes. This is the way you're remembering the point in your mind, right? Now, if you see the concept in detail, the I gene encodes a repressor protein responsible for regulating the lack of operon. Okay. So I gene encodes a repressor protein responsible for regulating the lack of operon. Z gene codes for beta galactosidase, which plays a crucial role in breaking the lactose into glucose and galactose that we already narrated for you, right? Okay, here the I gene makes a repressor protein means this repressor protein made up of how many polypeptide chain is not there in the slide. See, it's made up of 4 polypeptide chain. This repressor protein is made up of 4 polypeptide chain. Okay, if there is getting attachment to the Z gene can't do any expression. Y gene makes the permease enzyme. Y gene makes the permease enzyme. Okay, this permease enzyme makes a molecule entry into the cell and outside the cell. This permease enzyme makes a molecule entry into the cell and outside the cell. So, and then we are coming for all the 3 gene products which are essential for lactose metabolism. We know very well. Okay, here for the lactose metabolism to happen, not only structural gene, not only repressor gene, not only inductive gene, not only promoter, not only activator. In addition to all these things, we need the origin of replication. Origin of replication. So these all things are organized in the different places, right? Okay. So, for example, here we are going for one particular event to execute, right? At the time, dress code, mother is saying, this is the way you need to have a dress code. Father is saying, okay, this is the way you need to go in the transport system. Okay, here grandfather is saying, okay, here you need to worship the God and then you need to reach. Okay, like this, what many people are saying different information. Accordingly, what when I am going for one particular event, I am taking all their messages and doing everything. The same way, the lactose metabolism should be executed. Then we need the cluster of gene expression. The cluster of gene should work together. So the cluster of genes should work together. Then only what gene expression happens more appropriately, right? So now we are coming from the next basic concept in lack of prod is the repressor protein product produced consecutively by the IG binds to the operator region of the operator, right? So as I already told you, when the repressor protein will be formed, when the repressor protein will be formed, okay, you all need to understand the repressor protein will not allow the lack of prod to happen. This repressor protein is formed when the glucose is available. The glucose is available. Okay, when who will extract the repressor protein? Who will destroy the repressor protein? For example, I am the allolactose molecule. I will bind to the repressor protein and I will make the repressor protein to get disassembled. Disassembled. So you know very well, allolactose. Okay, here is responsible for breaking the repressor protein interaction with the structural gene. Right? Now, RNA polymerase enzyme it's very, very important to do the transcription process. Transcription process in the sense RNA production process. From whom the RNA production process will be initiated by the RNA polymerase from the DNA. Okay, who is doing this? RNA polymerase. What is the other name of RNA polymerase? DNA dependent RNA polymerase. So where it lands over exactly? It lands over the promoter. So is there is any specific RNA polymerase will bind to the promoter? Yes, of course, RNA polymerase 1. RNA polymerase 1 will bind to the promoter. So this RNA polymerase, after interacting to the promoter, immediately it causes unwinding of the double-stranded DNA. Unwinding of the double-stranded DNA, right? So what will happen is the glucose and galactose cannot serve as inducer for the lack of bond. Okay, inducer for the lack of bond. If that means what? Indirectly, you need to understand the glucose and galactose, which is an inactivator, which is a inhibitor, which makes the beta galactosidase enzyme, which makes the beta galactosidase enzyme not to work more appropriately, right? So this glucose and galactose is an issue. Now we are coming to the next phase explanation. Before going to the next phase explanation, the salient feature of the human genome, what we'll do is any one of our student is having any doubt in the lab account, please let me know. Before that, we'll discuss some questions. Okay. In 2023 March, this is the question we asked. Why is lac-operon said to be transcriptionally regulatory system? So what all things we got as of now? Everything we need to write topic by topic. A, B, C, D are all having different explanation, right? Can you see this please here? Lac-operon is transcriptional regulatory system because the control of the gene expression occurs at the level of transcription initiation, right? This is the way you record the message in your brain, right? So all the points be related subtopic wise, okay, for all the subtopic questions, right? Now we are coming for the next phase explanation. Question number 1 we discussed. Okay, this is subjective question. Okay, in the before 30 minutes back we discussed about the MCQ questions, right? So, what is the regulatory gene in lac-operon marked as I gene? Okay, this I gene regulates, of course. This I gene produces what? Repressor protein. When the I gene produces repressor protein, it produces repressor protein when there is a glucose availability. When there is a glucose availability. If there is a glucose availability, repressor protein will be not formed properly. If the repressor protein is not formed properly, then what will happen is glucose is there. If the glucose is there, repressor protein is formed. If the lactose is there, repressor protein will be not formed. Lactose, if lactose alone is there without glucose, repressor protein will be destructed. If only glucose is there or glucose with the lactose is there, then definitely what? The repressor protein will be formed and it won't allow the transcription to happen. It won't allow the gene expression to happen. Right? This is the way you are putting all the points in detail. Right? Now we are coming further. So I told about step by step each and every point how you need to explain everything in detail. Now we are coming for what are the levels at which gene expression can be regulated in eukaryote. If you're talking about, okay, here including transcription, transcription, translation, post-translation level. So, okay, here there is a way you need to understand A is the correct option for this, right? Okay, everything is happening in a periodical order. Transcription, post-transcription, translation, and post-translation is the periodical order. These ways, the gene expression happens in the eukaryote. Eukaryote means having well-established cell organelles. So, what happens in post-transcriptional modification? RNA is getting matured. What happens in the post-translation modification? Protein is getting matured. Protein is getting matured, right? So now we are entering into the question number 3. In the question number 3, what term is used to describe a group of genes with a common promoter and a regulatory gene in bacteria? So we know very well transposons, cisterns. The correct option is what? Operon. That we already narrated for, right? So the group of genes is controlling the gene expression. The group of genes controls the gene expression, right? So now we are entering the question number 4. The question number 4, which of the following is an example of an operon in bacteria? Here, in bacteria, Tryptophan operon is more common. Okay, TRP is tryptophan, is an aromatic amino acid. This aromatic amino acid, tryptophan operon, we are seeing the case of bacteria, right? So here, this we already told for you, right? Any one of you are having a doubt, please let me know. I'm there to help you. Now we are entering into the worked examples. Okay. So again, back and saying for you, the work the way you are seeing for all the things point by point, right? So point by point, we are narrating for the worked examples, right? So explain how the lack of protein is regulated in the presence and absence of lactose. So in the presence of lactose, of course, definitely lack of protein will be switched on, provided one condition no glucose should be there. If glucose is there, lack of protein will be not working. That we already told you, right? So the repressor protein will be formed by the lack of protein genes when there is a glucose availability. When there is a glucose availability, the lack of protein will be switched off. Describe the role of the transcription factor in eukaryotic gene regulation. Okay, eukaryotic gene regulation. Provide an example. Transcription factors are the protein that plays a crucial role in eukaryotic gene expression by influencing the transcription initiation. So here we all know very well. So the transcription initiation is controlled by sigma factor. Like this in the last biology class, we already told you, right? So the sigma factor makes the RNA polymerase to get landed appropriately in the case of promoter. So I'm RNA polymerase. If you need to get attached or adhered to the promoter, I need certain correcting agent. Who is that? The sigma factor. So the sigma factor is a part of what exactly RNA polymerase, right? So like this, what transcription factors. Okay, very good example, sigma factor. If you're talking about rho factor, it won't allow the transcription to happen furthermore. So that sigma factor, rho factor is not here in the slide. So we are coming to conclusion. You also should write the extra points for your better board preparation, right? Okay, now if you're talking about the gene regulation of insulin production in pancreatic cell. So in the presence of high glucose, obviously the presence of much amount of glucose, insulin production will be much more. If insulin is good, then only what the glucose amount will be reduced in the body, right? This is a message we need to understand. So transcription factors in eukaryotic gene regulation. So if glucose is much more, insulin production will be much more. If the glucose is less, insulin production will be less. Then the same way we told already to you, if the glucose is there, lack of protein will not happen. If the glucose is not there, lack of protein will happen. If the lactose is available, if the lactose is available, right? Now we are going for discussing the explain the concept of epistasis in the genetic gene interaction. See, irrespective of dominant gene or irrespective of recessive gene, if I'm epistatic gene, okay, that gene will suppress the expression of other gene. That will suppress the expression of other gene, right? Epistatic gene suppresses the expression of other gene. It won't allow the particular nearby allele to get expressed in the sense I'm an allele. I should supposed to make the RNA. I should supposed to make RNA, DNA, or protein. So all of the expression is completely blocked because of the epistatic condition. Okay, epistatic allele will not allow the expression of other allele. Either you're a dominant allele or either you're a recessive allele, whatever the condition it may be, I will suppress your expression. Like this, epistatic genes are narrating, right? So now, anybody are any doubt in this solution? I think so. No one is having a doubt in this place. So in all the examples we discussed, right? Subjective questions we already discussed, right? And then we are going for tips for the exam. Okay. See the lack of raw ingredients. Why lack of raw will happen? What is the benefit of lack of raw in life? In which way lack of raw is interrupted? In which way lack of raw is accelerated? So like this, if you put all these questions and then add the value points to that, so that what that will be a good narrative point for all of you, right? So now we are coming to the next important segment. Tips for the exam we discussed. Okay, examples we discussed. Okay, related videos. Whenever you have time, look into these videos, right? In the Gotouniversity portal.